Vitamin A's role in immune function is widely taught in basic nutrition education. It maintains epithelial barrier integrity, supports mucus production in the respiratory and gastrointestinal tracts, and is required for the development and differentiation of immune cells. What appears much less frequently in nutritional education is the mechanism connecting vitamin A to autophagy, specifically in immune cells, and why this connection makes vitamin A status relevant not just to surface-level immune defenses, but to the deep intracellular pathogen-killing capacity of macrophages and T cells. When vitamin A is converted in the body to retinoic acid, its biologically active transcription factor form, it binds to retinoic acid receptors in cell nuclei and directly regulates gene expression. Among the genes under retinoic acid's transcriptional control are BECLIN-1 and LC3, two of the most fundamental proteins in the autophagy machinery. BECLIN-1 initiates the autophagosome formation process. LC3 is the structural protein that forms the autophagosomal membrane and is universally used as a marker of autophagy activity in research. Vitamin A's regulation of these genes means it is not simply providing raw material for immune cells, it is controlling the expression of the cellular machinery that allows those cells to capture and degrade intracellular pathogens, a process called xenophagy that represents one of the immune system's most powerful strategies against bacteria and viruses that have found refuge inside host cells. This has practical significance for infection defense. Macrophages that have adequate vitamin A status can activate autophagy-dependent pathogen degradation more robustly than macrophages from vitamin A-deficient subjects, a finding observed in cell culture research and consistent with a well-documented clinical observation that vitamin A deficiency dramatically increases susceptibility to and severity of respiratory infections, measles, and gastrointestinal infections. The mechanistic pathway through autophagy gene regulation helps explain why vitamin A deficiency's immune consequences go beyond simply reduced immune cell numbers. The distinction between preformed retinol, found in liver, eggs, and dairy, and provitamin A carotenoids, found in carrots, sweet potatoes, and leafy greens, is essential for understanding how reliably these immune benefits can be obtained from different food patterns. Liver contains approximately 16,000 IU of preformed retinol per 3-ounce serving, representing the most biologically direct source. Provitamin A from plant foods requires enzymatic conversion by BCMO1, which varies from a 3:1 ratio in highly efficient converters to 28:1 in poor converters, with the latter producing minimal retinol from even large carotenoid intakes. Genetic variation in BCMO1 is a significant and underappreciated reason why some people on plant-based diets develop functional vitamin A deficiency despite eating abundant orange and yellow vegetables. A critical warning applies here with unambiguous clarity. Preformed vitamin A is the form associated with toxicity, and the upper tolerable intake of 10,000 IU daily should not be exceeded in chronic supplementation or through very frequent large liver consumption. Pregnancy represents the most serious context for this warning. Excessive preformed vitamin A is teratogenic, causing birth defects at intakes well below what some supplement products deliver. Pregnant women should work with a healthcare provider to ensure vitamin A status is adequate without exceeding safe limits.